So today we have a very important event, namely PhD defense of Anton Putintsev. Yeah? And let me explain you the procedure of the day. And first of all, I will shortly introduce the jury member. Then the candidate will have around 40 minutes of talk. Then there will be questions from the jury. After the questions of the jury, will not require, but we allow and encourage the questions from the audience. After that, the supervisor will have a chance to say some good words, I hope, about the candidate. And then the open session interrupts by a closed session where we will take our decision concerning the defense level and all that. And after that, I hope we'll disclose our decision and celebrate the candidates. Okay, this is the procedure. And let me, sorry? Yes, I hope so. You are very serious. I understand it. Yeah? So let's start from the introduction of the jury members. It is my great pleasure to be the chair of today's jury, and you see information about myself. And let me switch to our guests who kindly agreed to be jury members. And Simon Deliberata is a great scientist and our colleagues whom I know for a long time. He's renowned specialist in the topic of dissertation and polaritonic physics, and you can see on your screen the great trajectory of Simone through the science and through his career. And we really thank Professor Simon Deliberata to join our jury. Elena, can you show us the next? Daniel Sanvita, also I know him for quite a long time. We have contacted a lot on different conferences, and Simone's experience in different questions and domains of polaritonics certainly justifies his participation in our jury and put the level of the jury on very high level. I want also to express our great thanks to Professor Daniel Sanvita, who joined our jury. Next, please. Now we switch to our Skoltech professors and Professor Dmitry Gorin, who is with us already for a long time. He is a great expert in biophysics, biophotonics, and different applications of photonics aspects to biophysical and optical applications. And Dmitry is well-known specialist in this domain, and not only in this domain. So thank you very much, Dmitry, joining with us. I hope the next defense you will join us in person. Yeah? Great. Thank you very much. See you soon. Professor Sakelaris Miles, who is also a member of our team of School Center of Photonics and our center. And he is a great expert in modification of solid states by means of laser radiation. He studied a lot different structure formation, and we're also collaborating with him. And I'm sure that Professor Sakelaris will also strongly contribute to today's discussion and estimation of the level of our PhD candidate. Thank you very much. Next, please. Now we switch to the team who really made the job. Yeah, so Professor Pavlos Lagudakis, who is now the Vice President of Photonics Direction, and I think he do not need any recommendations, and he lead this work for already 4 years, as far as I understand, and probably also at the master stage.
No, the master stage I was in Moscow State University.
In Moscow State University, yes. Okay. But anyway, last 4 years, Professor Lagudakis was strongly involved in all this work, and we would like to, of course, thank him for this incredible contribution. Great. Now, as far as I know, yes, we should not forget the course supervisor who contributed, I think, a lot, even maybe more, because he was the nearest advisor of Anton. And Denis Sannikov, whom I know also for quite a long time, we also made some collaboration with him, and his level and his experience, I think, also show that. And we will see from today's work, I think, also the quite good, great level of scientific supervising. Great. And finally, we come to the main hero of our day, namely the candidate, Anton Putintsev, who graduated from Moscow State University and joined Skoltech at least 4 years ago, according to what I understand. Yes, and today, Anton will have 40 minutes for presenting his PhD thesis. And let us switch to Anton's presentation.
So thank you very much, Nikolai, for this introduction. And let me start. So once again.
Just a second, Anton, okay. Let me share your slides first.
Okay, so let me just check all the preparations and that my points works correctly, and I can switch the slides. So we can see. So those one who are joining us remotely, can you see my cursor on the slides? Please. Yes. Okay, then thank you. So once again, my name is Anton Putintsev. I'm a final year PhD student in the Hybrid Photonics Laboratory at Skolkovo Institute of Science and Technology. And today I would like to present you my work that I carried or conducted under the supervision of Professor Pavlos Lokoudakis and co-supervision of Dr. Denis Sannikov, titled Ambient Polaritonics. So my thesis consists of 5 main chapters. Starting with the introduction to and a brief history of the fields, where I discuss the basic physics underlying polaritonics in general, and give an overview on state-of-the-art organic materials or organic platforms suitable for ambient polaritonics. Then there are 3 consequent chapters related to the research itself, and that which make up the major part of my thesis. And I concluded with the discussion section. So let me, in my PhD study, I've been striving for solving challenges of, first of all, finding appropriate organic materials and demonstration of possibility to undergo quasi-steady state polarity and condensation that will be described in a lot of detail in my second chapter. Then I was striving for establishing pumping conditions and system parameters for maintaining such a regime, studied new designs of microcavities for implementing polarity and physics. So, and then I investigated the disorder and second order statistical properties of polarity and condensates in single shot regime excitation. So I also investigated how the size of the system affects parameters and main characteristics of the emission. And everything was done at room temperature. So I dealt with organics predominantly. Throughout my PhD, I have developed and exploited a broad skill set that includes both applying advanced experimental techniques in either conducting the experiment and various mathematical apparatus to analyze and interpret large amounts of data I've had to deal with and prepare results for publication in high impact and peer review journals. And so let me start with the first chapter, and I would like to give you an insight into how we differentiate between conventional photon lasing and polarity lasing. So on the example of a Ruby laser. So Ruby laser can be described as a 3-level system, which energy level structure you can see on the left of the slide. And in normal lasers, we can excite the system by means of optical or electrical pumping to excite the system to a higher energy state, from where, via non-radiative relaxation transitions, it can relax to metastable states, where it can live for up to milliseconds. From there, the system can spontaneously emit in all directions. But when the pumping rate is increased somewhat, we can create the inversion population on this metastable state with respect to the ground state. And first few spontaneously emitted photons, photons stimulate the chain reaction and result in a coherent stimulated emission from this metastable state. So the source of the coherent emission in this case is the stimulated emission into the photon mode. So here the system can emit to absorb from the photon mode. But however, if the strong coupling regime is realized in the system between semiconductor elementary excitations and cavity photon, or namely if the oscillator strength or energy exchange rate between energy exchange rate between those semiconductor excitons and cavity photons is larger than their decay rates, then new eigenstates of the system appear that we call exciton polaritons, partially light, partially matter states. And now the system, instead of having bare cavity and bare exciton modes, have new normal modes. In this case, upper polariton and lower polariton branches. And now, rather than emission in the system, we should discuss the scattering mechanisms from excitonic, high energy excitonic reservoir into the bright polaritonic states. Because once excitons have relaxed to polariton states, they simply decay from there. And this exciton reservoir, we can populate by pumping the system non-resonantly at a very high energy. Although we can inject polaritons directly into the system by pumping resonantly into the in resonance with the polariton states. So the relaxation processes in the system can be divided into two main subgroups. And the first subgroup is responsible for populating the polariton branches from high energy exciton reservoir. And these relaxation processes are different for inorganic semiconductors and for organic ones. If in the first one, the dissipation energy dissipates via high energy optical phonons and electron polariton scattering, then in the second group, in organic semiconductors, it is mainly via vibronically assisted radiative scattering or phonon-mediated non-radiative scattering. And now, once excitons have relaxed to polariton states, thermalization processes start to play a significant role, and they are also different for those two counterparts. So in the first, in inorganics, it is the interactions with acoustic phonons of rechargers or polariton scattering, while in organics, it is nonlinear interaction with low frequency vibrations. So once we increase the pumping in the system, more and more polaritons start to occupy the ground state. More polaritons start to occupy the ground state, up to the point when stimulated relaxation into the ground state starts to dominate over other relaxation channels in the system. And the buildup of a coherent microscopically occupied state occurs. This state can be described by a single macroscopic wave function where phase, momentum, energy, and polarization of the state are well defined. And due to the reason that polaritons have a finite lifetime, or due to the dissipative nature of our system, polaritons can decay. And photons that result from this decay can spontaneously escape the cavity. And these leaking photons or this leaking emission is actually a coherent emission because it results from the coherent state. And now in the system, we deal with a sort of spontaneous emission from the coherent state, which is in fact coherent. This emission inherits those properties, the properties of this state, and in particular the momentum, energy, polarization, and phase. And detecting this emission, we can get an insight into the processes and physics that happens inside the cavity. So this is how the dispersion or condensation process looks like in the experiment and in our system. And I don't know if I can play the video because, Yelena, can you help me or assist me here? Because this is the video, yes. So the hallmarks of the condensation you can see on the left video. So on the top right figure, you can see how the intensity, or which is proportional to the polariton population, depends on the energy on the lower polariton branch. To the bottom right, you can see the ground state emission, and on the left bottom figure, you can see how the blue shift of the ground state emission evolves upon increasing the pump power, and also how the line width of this emission evolves. So we see that upon increasing the pump power, we observe the nonlinear increase of photoluminescence, polariton photoluminescence from ground state. We observe the step-like behavior in blue shift, and we also observe the build-up of a temporal coherence as indicated by the drastic narrowing of the line width of the emission. But those hallmarks of the polariton condensation that are usually attributed to it are not in fact sufficient to justify or to claim that the system really has polariton lasing, because it doesn't verify. So we need to do the complementary measurements to verify that the system indeed is in strong coupling regime. And for that, supplementary or complementary measurements of anti-crossing between polariton branches are needed. So, and those measurements are usually, so in principle, reflectivity measurements at high angle, where you can see the Rabi splitting between the branches. So far, polariton condensation has been observed in a wide spectrum of organic materials. It was seen in anthracene organic single crystals, in the family of fluorescent molecular dyes, then also conjugated polymers that to some degree combine the properties of organic single crystals and fluorescent molecular dyes. Also, the polariton condensation has been seen in green fluorescent proteins, and this work was quite remarkable because those proteins can be biologically derived, and this is really remarkable to me. And recently, the polariton condensation has been shown in the family of perovskites. So in all inorganic and 2D layered iodide perovskites and organic and inorganic chloride perovskites. And this is in this work, the authors show really peculiar effects and behavior, which was previously possible only and were seen in inorganic semiconductors. And this work practically eliminates the gap between inorganic and organics, because it was done completely at room temperature. That's why, in particular, the optically induced lattices of polariton condensates, the interaction between two spots with the clear fringes in between, they were observed, and everything was shown. And in my work, my research has been predominantly focused on those two materials, so in brominated fluorescent molecular dye and on conjugated polymer. So these two guys I studied in great detail. So throughout the PhD in order to conduct all those experiments, I designed and assembled the setup that one can use in different configurations, and it was fully automated. So one of the important configurations that it can work at is the bulk or single shot detection and acquisition of photoluminescence under a variable pulsed excitation. So under pulsed excitation with a variable length, so from 300 femtoseconds up to 10 picoseconds. And it allows one to do a single shot measurement or detection of real space and K-space emission, which is also can be resolved in polarization. By bulk, I mean the detection of pulse trains, and each characteristics of each pulse in the train can be detected separately. Yes. And also, this setup allows one to do a single-shot pump probe scanning with 3 independent pump pulses. So one pulse is fixed at 400 nanometers, the second at 800 nanometers, and the third one can be spectrally tuned in this spectral range from 420 up to 800 nanometers. And this actually allows one to study a lot of interesting physics and effects. For example, you can simultaneously create the population in different organic materials in a multi-layered structure in order to study the relaxation dynamics or other interesting effects. But this part of the talk will, so I will not talk about this part because it was not included in my thesis. So let's start with the second chapter, and the motivation for this part was that first of all organic materials, they tend to photobleaching, and they degrade under intense optical excitation, and hence none of them can sustain a CW or continuous wave excitation. That's why we need the pulsed operation. And also, it is really important for organic polariton condensates to we need to extend their lifetime in order to observe or give them time to interact with each other, because intrinsic lifetime of polaritons is known to be very short, and it is of the order of hundreds of picoseconds. And since the nature of the excitons in organics is different from inorganics one, then these 300 picoseconds or hundreds of picoseconds is not enough for them to interact. That's why we need to create conditions and maintain them in order to let them interact. And in this work, and here I studied the brominated fluorescent dye, fluorescent molecular dyes. And first of all, I did the basic characterization of those cavities. So these were conventional DBR cavities with this active material placed in between. And on this experiment and in this work, I really tested and the designed a single-shot excitation setup with full automation of detection different parameters. And I studied, and what we observed that the system under the nanosecond or 6 nanosecond pulsed excitation can really undergo the polariton condensation. So as you can see on the figure, it goes from the linear regime to nonlinear regime from inset B to C, and how the condensation looks like in dispersion images with the hallmarks as nonlinear increase of the intensity of polariton emission, drop of the line widths, and increase in blue shift. But the question was, even though it is caused by a nanosecond excitation, whether it lives for nanoseconds. And that's why single-shot time-resolved photoluminescence measurements were carried out. So here you can see 2 figures of data collected in the linear regime and in non-linear regime. So first of all, the IRF of the detection system was measured, and it happened to be 1 nanosecond. And then on the top left figure, you can see with the blue dashed line the time profile of the pump pulse, and with the red solid line the time profile of the photoluminescence, and which is 500 picoseconds longer than the pump pulse, which is actually due to the exciton dynamics. And this is a known fact that excitons in this kind of material lives for on the order of nanoseconds, half or longer, depending on the concentration of the solution, percentage of those molecules in the solution where it was prepared. And once it goes to the condensation regime, we observe the so on the bottom figure, you can also see with the blue dashed line the pump pulse, which was of around 5 picoseconds, and 2 curves that represent the actually measured time profile of the photoluminescence. And it's the convoluted version with the IRF of the system, which happened to be 1.8 nanoseconds. So this scale, this nanosecond scale, is actually a quasi-steady state regime for the system where the polarity of where the intrinsic polarity in lifetime has around 150 femtoseconds. Yes, and the main takeaway of this work was that the molecular dyes can exhibit long life polarity and condensation, which gives a promise for these materials to study the interaction between multiple condensates. But this material is extremely prone to photodegradation. That's why this part needs to be really developed, because it can sustain a couple of pulses and we need to move to another position on the sample. That's really sophisticates the measurements and the work with this kind of a material. And the results of this work were published in Applied Physical Letters. Now we move to the third part, to the third chapter of my thesis, where I studied novel or new designs of the cavities for polariton applications, and it is called the hybrid metal dBr organic microcavities. So the hybridity here lies in the fact that instead of using just conventional set of pairs, dBr pairs as a bottom or top mirrors, a substrate of argentum mirror was used with a variable number of pairs of dBrs on top of this substrate. The question was whether such a system can, whether it is possible to create such system with the parameters qualitatively or sufficient for polarity and physics to be observed in this system. And the question, so for that, the active medium in these cavities was chosen to be bromated fluorescent molecular dyes among all this spectrum of known organic materials. And the setup in reflection configuration was also assembled. So I used the one objective to excite the cavity and the same objective to collect photoluminescence. So, and what was shown is that indeed we can fabricate those cavities with strong coupling regime, as evidenced by the reflectivity measurements on the top left figure. The quality factor or Rabi splitting also depends on how many pairs of those dBrs we use on top of the substrate, on top of the argentum substrate. And we observed that the system can really undergo polarity and condensation under a single shot excitation regime, and the parameters of those of this condensation of this emission was compared to the conventional or controlled dBr cavities with approximately the same parameters and the same thickness of the active medium. And it was shown that so the main takeaways of this work were that those cavities indeed are really easier to fabricate, because the less number of dBr pairs you use, the less you are prone to the errors on the stage of the fabrication. Those cavities exhibit quality factor of the system and sample homogeneity of the order, which is quite the same with conventional dBr cavities, and they are suitable for realizing polarity in physics. And this work was published in scientific reports. And now what we observed also in those cavities is that the emission in real space and either in K-space is fragmented. So in this work, it was attributed to the inhomogeneity and the thickness variation of the active medium inside due to on the stage of the fabrication process, which led to like non-linear landscape, potential landscape of the medium, which resulted in non-uniform gain medium, and that's why we observed the clusterization or filamentation of the condensate in real space, and either in K-space, as I said before. And we decided to study this fragmentation in the more details. And for that purpose, we chose a more photostable polymer, which was conjugated polymer mil-PPP. And this is the motivation. So this is how the condensation looks like in single shot in this system. On the bottom row, you can see single shot realizations for different spot sizes. And you can see the high degree of fragmentation or filamentation in the system. And on the bottom, you can see how the picture looks like when we average our picture over many realizations and over many positions on the sample. And the shape tends to replicate the Gaussian profile of the pump pulse. So initially, this system was studied at very different angles. It was studied in the transmission configuration. So it was detected from excited from one side and detected from the other. And the schematics, principal schematics, you can see on this slide. So, as I said, the system was studied at different angles. And first of all, it was studied on the question of static disorder, where on the same position on the sample, a lot of images of single shot realizations were collected and then averaged. And the degree of its deviation from the ideal Gaussian 2D profile was extracted. So this is how the map A minus B looks like. Then it was studied on the question of stochastic disorder, when the deviation of two adjacent consequent single shot realizations on the same spot with the same under the same excitation conditions and with the same pump profile parameters were studied. So it was so here, for example, so eventually it is how one realization of the condensate differs from the previous one, and so on and so forth. So stochastic disorder. And also those sets of single shot realizations were studied by applying the Pearson correlation analysis. And we were trying to find some correlations between the particular filaments or particular spots on the special profile. And this work actually was really sophisticated and meticulous, and it involved, so the interpretation of the result would involve some biophysics and some extra analysis that went beyond the scope of this work. That's why this part deserves the additional, like I would say, report, and it was not included in this in the thesis. But the results were quite interesting, because some correlations between particular filaments upon increasing or decreasing the pump power, or depending on how far above the threshold you are, were revealed. So yes, once you go higher and higher above threshold, so anti-correlation regions became correlation regions, and so such kind of dependencies. But this is another talk. I will not talk about it anymore. And I will predominantly focus on this part. So first of all, we decided to study second order coherent properties. We decided to start with it. And for that, I applied the bulk acquisition and at 10 Hertz repetition rate. And for each set of parameters, I collected 300 single shot realizations. So each of the single shot realization then was integrated, and the map, so on the right figure, you can see how, for example, the distribution of intensities or integral counts on the single shot depends on the pump power. So how it depends on how far above threshold you are. So with a different color, you can see how the histograms look like. And you see that upon increasing the pump power, the distribution actually looks much narrower than it was above threshold. And what those distributions what information those distributions can give us is, first of all, the average intensity or average polarity and population at these particular excitation parameters, and also the width of the distribution. So these two parameters are very valuable because they can be used to define second-order coherent properties of the system. So second-order coherence function can be expressed in terms of the intensity, so in terms of the photon number, and it can be it can be written down in the form of exactly those parameters. So the width of the or dispersion of the distribution and the average number of polariton population here. And this is how the dependence of this G2 function at 0 looks like with respect to the total polariton populations. And as we know from, as it is known for second order coherent light matter waves, this G2 value at 0 equals 1. And when for strongly fluctuating, for example, emitters, G2 at 0 exceeds 1. And below threshold, exceeds 1, and below threshold for thermal source, it is equals 2. And on this figure, there are two interesting regions and two important regions. So the first region is below threshold, and the second region is above threshold. Below threshold, the data cannot be really trusted because there are several reasons why we do not observe G2 values at 0 equals 2. So first of all, the important parameters that defines whether we can resolve this dynamics is the intensity correlation time and time resolution of our detection system. And below threshold, time resolution of our system actually exceeds intensity correlation time. And that's why different statistically independent fluctuations, they are averaged. And also before threshold, there are a lot of modes that are occupied on the bottom polariton branch. That's why a mode selection becomes inadequate. And as I said, statistically independent fluctuations from different modes are averaged, and this actually falsifies the measurements of this data. But above threshold, we see. But above threshold, the situation changes completely, and actually, I will show on the next slide. So the main takeaway from this slide that for at 5 thresholds above polariton condensation threshold, we can reach G2 value at 0 of this value, which is actually very low and is only 100 times above short noise limit for such system. So for such total population number of polaritons. And so for a single mode polariton laser, the functional form of the G2 function is actually as that of the normal lasers, and it can be expressed as follows. And if we now average this G2 function over the time of the measurement or time resolution of our system, we can connect what we measure in the experiment with the actual G2 values that the system has. And you can see that it is, so what we measure is strongly dependent on the intensity correlation time and time resolution of our system. So, and this intensity correlation time is different, is defined differently for the cases below threshold and for the cases above threshold. So for the case below threshold, it can be defined by just simply measuring the line widths of the polariton emission in ground state, which happens to be 277 femtoseconds. And above threshold, it depends on the gain saturation number NS, and also on the polariton population and on the decay rate. And time resolution in our case is actually condensation lifetime, because when we measure single shot realizations, we integrate in the window on our EMCCD camera, we integrate over 5 milliseconds, but these 5 milliseconds encompass the whole photoluminescence that was emitted under pulsed excitation. And actually the condensation lifetime was assumed in this experiment to be of the order of 1 picosecond or 2 picoseconds, which was also actually verified in our different experiments, which are currently in process or ongoing experiments. And so yes, and above threshold, the situation is the following: that the resolution time of our experiment is much shorter than the intensity correlation time, and that's why what we measure in the experiment asymptotically reaches the actual G2 values at 0. And what we decided to do further is, on the grounds of the previously published work, it was known that changing the size of the system can really affect the degree of filamentation of the system. And we decided to check whether changing the size of the system can affect our coherent properties. And that's what WEOI carried out. So I did the spot size dependence and how it affects really the real space profiles, how it affects K-space profiles, how it affects G2 at 0, and also how it affects the integral characteristics of the emission. So first, it was observed that under the same excitation conditions, so namely once we change the excitation power, excitation fluency, but we're still, once we change it, but still stay at 2 thresholds above condensation threshold, which is different for different sizes of the condensate. We can observe that the fluctuations of the intensity fluctuations of our condensate, and hence the G2 value at 0, starts to increase. And we also observed that the slope of the nonlinear region or transient region also changes. And also what changes? The threshold of the polarity and condensation also changes. We tried to, we developed a simple model, a first approximation model that can, first of all, explain the behavior of the threshold. So we introduced additional term in the decay rate of the polaritons. And this guy, actually here, gamma out, is attributed to the kinetic losses of the polaritons. So polaritons, they spread, they have non-zero group velocity, and they tend to escape from the condensate. So since we pump with a Gaussian profile, there is a non-zero gradient, and polaritons have an outflow with a particular group velocity that depends actually on the size of the condensate. And this functional form of the threshold with respect to the size of the condensate was derived, and we fitted our threshold dependence on the spot size with this functional form, and we found sort of a good agreement between theory and the experiment. And this was also used to explain how those kinetic losses affect the G2 values. So as you can see, once for the big spot sizes, the contribution of the kinetic losses into the threshold or into the fluctuations, it can be neglected. So, for example, here in particular, for starting with 40 micron size, and here starting with 30 or approximately 40 micron size as well, there is no difference for the condensate of what size it is. So that's why the contribution of kinetic losses here is negligible. Okay, and also in order to verify that the analysis of our G2 values and overall experiment was correct, the dependence on the technical analysis on the how G2 value depends on the region of interest over which we integrate the total counts on our EMCCD camera was carried out. And as you can see on the left figure, that starting with a particular or in particular 20 micron diameter size inside which we integrate total intensity, the contribution of the EMCCD camera noise doesn't really affect the total intensity fluctuations of our system. And this figure is really important because it shows that what we observe here upon reducing the size of the system is really attributed to the intrinsic noise of our condensates. And also on the right figure, you can see how the fluctuations of the pump laser look like. How the fluctuations of the pump laser affect the statistics. And actually, it doesn't affect statistics very much, because you see that the functional dependence is quite the same. So what I mean by what analysis was carried out here in particular is we took all 300 realizations, and since we know the pump pulse or pump power for each of the realization, they were filtered within a different region of pump powers. So with a tolerance interval of 1%, 2%, 5%, and 10%. And what we observed is that this increase of G2 values that we observe is not really affected by the the fluctuations of the pump laser. And this is actually a very also important result. And so the main results and takeaways of this work is that an alternative approach to access second-order coherence that is based on well-controlled single-shot detection was carried out. And it was demonstrated that highly disordered organic polariton systems can reach an extremely high second-order coherent emission with the record values. So, or not the record values, but first. I would say firstly reported values in organic polaritonics, because so far there were no experiments that were carried out in this domain. So to measure second order coherent properties in organic polaritonics. Also, the nonlinear polariton system behavior was studied, and how it depends on the reducing of literal size of the system, which was also supported by microscopic model and describes system dynamics. And the study of the stochastic and static disorder contributions to spatial filamentation was also shown. So this is, I think, that's it for the main results. And so these three works were mainly these three works make up my thesis, although I have other ongoing projects that were not part of the thesis yet. But yes, and this thesis covers these three projects. So thank you for your attention, and I would also like to acknowledge the contribution in one way or another all my colleagues from the lab. Not all my colleagues, but some of the colleagues. Not all of them were included, actually. I think all of them were. But so the greatest acknowledgement is to my supervisor, Professor Paulos Logodakis, who really was really supportive and really made possible all those experiments with all these resources and all his guidance throughout all 4 years of my PhD. Also, I acknowledge the Dr. Anton, who made a really great contribution to my works, and with whom we had really fruitful discussions, like analyzing and interpreting results. To Dr. Denis Sannikov, who was actually side by side with me all those 4 years, and with whom we stayed late nights and did many experiments in the evening, and with also fruitful and long discussions upon the results and what should be carried out further. And to the rest of the team. So yes, so I'd like to thank them all, and thank you for your attention once again. and I'm open to your questions now.
Thank you very much, Anton, for very good, interesting presentation and perfect timing, 40 minutes exactly. Yeah? So now we are switching to the section of questions of jury members. And after that, I'll ask the audience also to ask the questions. So, dear colleagues, dear jury members, are there any volunteers to start? We all are free, so I would propose the word I introduced you, but it's absolutely not necessary. If someone would like to start, you're really welcome.
Okay, I'll start with the question.
Timone, you are really welcome. It's you.
Yep. Well, already, thank you very much for the interesting manuscript and for the very clear presentation. I have a few questions. The first one, what is the origin, the microscopic origin of this filamentation?
Microscopic origin of this filamentation.
Yes.
So there are several reasons why we observe the filamentation in two particular cases: in averaged picture of the condensation and then single shot realization. So, because we clearly observe a static pattern which can be seen while we average, for example, 300 realization stain on the particular spot, and I attribute it to the inhomogeneity and disorder of the active medium inside, because when it is spin coated, there are some clusterization of the active material inside, and that's why the gain of the medium differs from position to position. That's why when you excite the system, there will be clearly some areas with a higher gain and some areas with a lower gain. But it doesn't mean that the condensation doesn't occur in between. It's just not that intense as on the particular filaments. But what we also observe is that from realization to realization, there is a stochastic pattern that doesn't repeat itself completely. So it means that there is some dynamics and instabilities that cause such peculiar behavior. And this behavior was studied in different systems by many researchers, in particular by Bobrovskaya, in works by Daskalakis, and there it was, and depending on whether this filamentation happens in organic or inorganic materials, the underlying nature is also different. So there is one of the reasons there is a hole burning effect when the gain in the particular regions is suppressed, and it really involves the interplay between time dynamics of Bogolyubov instability spectrum and other things. So I do not really recall completely what were the times, the characteristic times, but they really depend on the size of the system, because when you decrease the size of the system, you cannot reach this instability, and you observe sort of a single formed condensate. But once the condensate is bigger and bigger, more domains start to form. And but in inorganics, it is predominantly the disorder of the active material inside.
Okay, thank you. Can you, on the same line, can you comment on how this filamentation affects the coherence and the nonlinear properties of the condensate?
How this filamentation affects coherent and nonlinear properties of the system?
Yes.
Okay, so in this work, I didn't study first order coherence of the system, because in the previous work by Bobrovskaya, it was shown that in the interferometry measurements, they demonstrated that under the Gaussian, under the same excitation conditions that we carried out in this work, they observed a uniform interference pattern without any dislocations in fringes. That means that different filaments have the same phase, and which means that different filaments are part of one big condensate. But they also demonstrated that if they go from the Gaussian pump profile to the top hat profile, they can observe the situation when there are dislocations in interference pattern between interference fringes. That means that some filaments start to form with different phase, and it actually not so. And this is a slight indication into the fact that those filaments can be just parts of different condensates, which are not coherently coupled to each other. But in this, in our work, first of all, the line width of our condensate is really narrow. Even though within the line width we can observe, even in K space, small filamentation or small fragmentation in the dispersion single shot measurements. But still those filaments lie within lie within the line widths of the emission. That's why we can say that they, first of all, coherent. So they have the same energy, and they are coherent to each other, so that they have a temporal coherence. But regarding the spatial coherence, so when I did the interference measurements, I observed that there were no dislocations in between the interference fringes, but I didn't study it in a very great detail. What I can say about how the coherent properties changes when we reduce the size of the system. So first of all, the stochastic pattern or stochastic filamentation that we observe in our condensates, it doesn't really change, because the degree of the stochastic disorder doesn't change when we reduce the system size. That's what we observed in this work, in contrast to the previously reported works. Yes, this affected the second order coherent properties of the system, as you saw on the dependencies that I showed previously. But for the first order coherence, it is still, I believe, and this is all filaments are part of one condensate. But this requires so to know for sure, that requires a very, I would say, particular measurements, and we need to discuss. We need to study it in a more detail. And regarding how filaments affect the nonlinear properties of our system, so I did not study how a particular filament affects on the particular dependence, on the particular characteristics, or on the particular integral characteristics of my emission. I observed the filamentation of the whole condensate, and I studied the sort of integral characteristics, the integral intensity, which fluctuated from shot to shot, and how the S-curve or pump power dependence of the intensity behaves. We observed that so upon reducing, we can change the slope of the transient region from thermal to coherent state. So we can slightly change. Upon reducing, the slope becomes steeper. But since filamentation doesn't change with changing the size of the system, it is, I don't think it's, we can really talk how it affects, it doesn't affect really the nonlinear properties, because filamentation itself doesn't change when we change the system size.
Okay, thank you. Other question, could you comment about the link between the hybrid DBR structure and the existence of TAM plasmons in that kind of structure.
Sorry, the link between hybrid structures and
TAM plasmons are those excitations between a reflective surface and a DBR. Is this somehow linked to the, or can be observed, or is it present in the hybrid resonators you have been considering?
So actually, it is an interesting question about TAM plasmons, because I did not really look into this question throughout my research. That's why so it's sort of difficult for me to pop out the answer from my head.
If you didn't study that. Okay, can you go to slide a few minor questions on some slides? Can you go to slide 14, please?
Elena, can you help me to get my slides back? Yes, the slide number 14. Yeah.
I don't see it on the.
Because it's not sharing right now, so we will share in a moment. Okay, so I will just press. Yeah, this one.
Okay. Can you comment a bit more in detail the change between panel A and panel C? Explain a bit better what the underlying physics, what's happening in the underlying physics when you see the change in the PL time between the below and above threshold cases, please.
So no, first of all, okay, when we are below threshold, what we really examine and what dynamics we deal with is actually the exciton dynamics of our fluorescent dyes. Because when we excite the hot exciton reservoir, so there are different relaxation channels in the system, and since the threshold is not reached, the time dynamics of the red curve, which you can see, yes, on panel A, actually follows the exciton dynamics, exciton reservoir dynamics. And here, with the blue dashed line, you can see the time profile of our pump pulse, which is of order 5, so which is of 5 picosecond width. When the system is pumped, we have a constant supply of excitons in our exciton reservoir. That's why we can observe the emission throughout all the time when the pump is active. But when the pump starts to decrease, we can see that there is a small delay between the photoluminescence and the pump pulse. And this delay is actually of the order of 500 picoseconds, which means that if we, for example, would excite our system with a delta function, we just put the population of excitons in the system, then we would observe the exponential decay of our photoluminescence, which would be of 500 picosecond length. And this is the exciton dynamics. But once we go above threshold, and the panel C shows how the photoluminescence shows the photoluminescence dynamics in the in the above condensation regime. The stimulated relaxation into the ground polariton state starts to dominate. And the fast depletion of the hot exciton reservoir happens. It means that upon increasing the pump pulse, which is depicted with a blue dashed line, we see that in the beginning there is no condensation, there is no nonlinear increase of the photoluminescence, because the threshold at this particular time is not reached yet. But when the threshold is reached, we can see how we can see the buildup of the coherent state of the coherent ground state, from where we can see a nonlinear increase of the photoluminescence. So it increases upon increasing the pump further, upon increasing the power of the so within the pulse time, and then it reaches its maximum. And we can see that the photoluminescence or polarity and condensation lifetime does not replicate. A small remark that what we actually measure in the experiment is depicted with the red dashed line here. So you can see red dashed line. It is slightly, sorry, it is slightly wider than the red solid line, because the solid line is was extracted in post-processing when we deconvoluted the signal that we receive with the IRF function of the system. That's why it's a bit narrower. So we will follow the we will follow the red solid line, and it doesn't replicate the pump pulse. And it means that the condensation at some point start to either quench for some reasons, because probably there is the photo bleaching in the system. It is known fact, and there is not enough. So strongly coupled molecules in the system, they become not photoactive, and we cannot provide enough enough excitations in our active medium to be able to replenish the condensation in the ground state. And yes, and here we see the rapid depletion of the exciton reservoir as a result of the rapid decrease of photoluminescence intensity in the condensation. And this process takes around 2 nanoseconds. So even though the pump pulse was around 5 or 6 nanoseconds. And also the fact that at some point the pump pulse becomes lower threshold, which can be drawn as a straight line. If we draw a vertical line starting with a nonlinear increase of the PL, we can see the point of the presumably of the threshold power of the pulse. And here on the other side is somewhere when the threshold is we go below threshold. So this part is also sort of below threshold.
Okay,
but
so, sorry, just to understand, if at 6 nanoseconds I would switch back my pump at the same level, at the maximum level, which now you reach at 2 nanoseconds. What would happen? So if the blue dash line at 6 nanoseconds at a second peak at the same height at the one it had at 2 nanoseconds, would the red line do?
So this is actually, for example, if instead of, I will answer in 2 parts. So if instead of this form of the pulse, we would have, for example, a rectangular pulse, ideal. So it reaches its maximum here at 2 nanoseconds and then goes straight, like a straight line in time domain. I would expect, and that's what we saw, that the photoluminescence doesn't follow these dynamics, doesn't follow the pump pulse. It means that some photobleaching happens in the material, and it needs some time to recover. So it means that there is some characteristic time which describes this photobleaching or recovery from photobleaching, and this dynamics was not studied by me. And depending on the time scale, the answer to a question will be different. So, for example, if the photobleaching exceeds like the order of 10 nanoseconds and more, so the recovery time from the photobleaching, it means that if we put at 6 nanoseconds the second peak, we will not observe the second condensation or the second nonlinear increase in photoluminescence. But if the recovery time is much faster, then
I got it. Just another point to really clarify my understanding of this figure. We see here both the pump and the condensate, which peak at the same time. But from what you are saying, it seems to me that you are saying that the peaking of the condensate there is just an accident due to photobleaching. What would have happened if the blue line had kept increasing till like 4 nanoseconds? So you would still dial up the power, not stopping at 2 nanoseconds, but going all up till 4 nanoseconds. So at 4 nanoseconds, of course, the power would be larger. In that case, would the red line still peak where it does at 2 nanoseconds, or it would peak at the maximal intensity at 4 nanoseconds in the example I did? I would like to understand if the coincidence of maximal of the extremal point for the red and the blue line are a coincidence, or if there is an underlying physics which links them.
So actually, the blue line and the red line were recorded on they were recorded on the same like on the same detection on the same oscilloscope, but there is a time lag or time delay between those two measurements. And in order to represent those figures, it was so the peaks or extreme points were matched in the analysis, and that's how we this is how I understand and how we agreed to understand the dynamics or physics that happens here. So that the peak intensity of the photoluminescence reaches the peak intensity of the pump pulse. But and also and this is actually on this slide, so on the representation of this figure, it is slightly speculative to say that where exactly should be the extreme point of the photoluminescence with respect to the extreme point of the pump pulse, because you can artificially shift it to the left or shift it to the right, but
you put them at the same point. So this is an information you are giving me in the figure, and so I don't understand that completely. So I'm slightly uneasy about this figure. So you are telling me that it could have been plotted differently.
So it could be, I would say, plotted differently. But the main conclusion, or the main the main conclusion that we can draw from that was drawn from this experiment is that the lifetime of the lasing is shorter than the pump pulse, and it doesn't replicate, it doesn't live for like a CW polariton lasing. And actually, if the extreme points they do not coincide, it doesn't really in this term affects the results of this work, because it can be shifted. It can be the peak of the photoluminescence may happen in time earlier than the peak of the pump pulse, and then, for example, the photobleaching happens, and the photoluminescence reached, for example, or the photoluminescence reached the saturation point. It doesn't grow further, or just the photobleaching happens and it just starts to decrease. But yes, so my answer would be that we could speculate about whether those extreme points coincide with each other.
Okay. Other question. Can we go to slide 26, please?
36?
26.
26. Okay, yes. Okay.
Okay. I'm trying to understand a bit better that can you explain to me the actual the equation, the main equation? Can you explain this to me, please?
Which one?
The one with the dashed red line.
With the dashed red line. Okay. So the functional form of the second order coherence time, so we can write it in this form. And if we assume that the pulse, we have an exponential pulse that has a width at 1 over e level of this value, we can average, we can write down this integral, which is the average over the in time domain over the delta t, and that's how we find, in fact, the so the value, so the G2 average, the G2 value at 0, which is expressed in terms of the G2 value at 0 actual one, so this one. And
Sorry, I'm trying to understand where where does the tau c comes out?
when does the tau c comes
Yeah,
out? So
from the second step of the equation to the third one.
Sorry. Okay, it comes when we find the so the pulse. So the delta t is actually the time resolution of our system, so it stays here as normalization constant. But since our G2 value or G2 function has a time dynamics which is described by this constant, if you find, so it means that the integration is not actually from 0 to infinity. So we integrate from 0 to time c, tau c. And if we find what is the value of this integral, it can be converged to this form, actually.
Okay, fine. So the central integral is not to infinite. Okay, great.
Yes, yes.
Can you go to the following slide, 27?
Following slide, okay. Yes. This one.
Yes, no, okay, it's fine now. It's yeah, okay. Last question. Can you comment a bit on the dependency of the dynamical loss term on the geometry, on the geometrical parameters? This is
So sorry, the dependence of dynamical
of the dynamical loss term on the geometrical parameters of the on DLS.
D and S, D and C, you mean?
Yeah, I mean, you are there. You are saying that is a dynamical loss term. Essentially, you create polarisons and then they escape. So this you said that this depends on the waste size, on the size of the condensate. I just wanted to ask you to comment on how this loss rate depends on the geometric feature of the condensate. I see there you have geometric dependencies, and you said that you have geometric dependencies. Could you please explicit that a bit?
Sorry, can I repeat your questions or question that, so in order to like clarify that I understand it correctly? You're asking about panels D and C, right?
No, I'm not saying. I'm asking about gamma, the actual gamma out.
Gamma, how this dependence, how this functional form was derived, right?
Yes.
So the polarity and decay has so we decided to introduce, so there are two assumptions or hypotheses that we introduced into our system, that the polarity and decay consists of two main two main facts contribute to the total polarity and losses in the system. First, it's just the natural polarity and decay time, which is denoted with gamma naught here, and the gamma out. So actually, if we just assume that those small dots or like polaritons inside the condensate they have a group velocity, and it means that they have an outflow from the center of the region, which is depicted with a dashed circumference. We can write down actually this sort of flow of polaritons through the border of this circumference. So it depends on the lateral size, on the length of this circle, and it also depends on the total area of the pumped condensate. So since the area is proportional to the square of the diameter, and since the perimeter or the length of the circumference linearly proportional to the diameter of this area, so we are left with just the radial parameter in the
Great. I agree on that. I just normally those are typical loss time, which depends on 1/r. I was a bit surprised to how it has been put as a surface over diameter, but now it's clear. Thank you. With this, I'm done with my questions. Daniele.
Okay.
Thank you very much,
Thank you
Simona,
very
for your
much,
questions.
Simona, for a very detailed discussion.
Thank you.
And
okay, first of all, let me say I really like, of course, this work.
Just kindly ask each of you member to confirm that you are satisfied with the modifications of the thesis made by Anton.
Yes, exactly. I was coming to that. So I really like.
I am satisfied.
Okay. The work done, and I'm sure it was really
great. So I ask each jury members during the questions he is performing also to make the answer on this my formal question. Okay, let us go further, and now let's pass the word to Danielle, if you are not mind. Danielle,
sure.
Yes, you're welcome. Really welcome.
Do you hear me?
Yes, perfectly. Danielle.
Okay, good. So let me say that I like the work, and also I think that it's been done with a lot of passion, at least experimentally. And I mean, maybe what I actually noticed was that it was a bit written in a rush, at least the first part of the thesis, but then the correction, I think, were well done. So I'm satisfied with the second version of the thesis. Okay, so let me ask a few questions, but I think some actually are curiosity that I would like to somehow also understand. Maybe we can start from the last slide, given that it was already there before. I will go a bit jumping from one to the other. Can you put it back?
No, it's not the last one. Those were
the last what was discussed.
Slide number 30. I guess 30. Yes. Can you go up?
Maybe
29.
27, something like that. Around 27, I think.
Upper. Slide number 28. Yes. No, 20. Next one. Okay, I'll manage from there.
But I should see it. We actually don't see in the screen.
Can you share the presentation, and I will manage from here, from there. Yep. So this one.
Not before, I think 27, wasn't it? Well, the one in which you were discussing exactly the dependence on the group velocity of the threshold.
This one.
Okay, so this is of course in hypothesis, right? So I would like to understand a bit better, because to be honest, I mean, I know in organic semiconductor, you see really clearly this expansion. So there is a strong blue shift, and given that all the states outside have a lower energy, so there is a very easy way of escaping from the spot by ejecting polariton into high momenta and so high group velocity state. Now, to be honest, so from both the real space image and also from the dispersion, I'm not sure I can see this effect. So even in the smallest spot, the dispersion seems to have a slightly higher K from K0. First of all, it's not symmetric, so it would be interesting to know why there is this asymmetry. The same asymmetry seems to be also for a bigger spot. While all this filamentation seems to be random, the direction of K is mostly in one direction, but this could be given by a wedge. I'm not sure if this is the case. Yet, I would like to know a bit better from what kind of experimental evidence you could work out that there is a strong gamma out, which is given by this expulsion of polaritons.
Okay, thank you for your question. Actually, so I would first of all be very accurate with the word strong outflow, because I have not included the corresponding figure in the presentation, but you can find it in the written copy of the thesis, where there is plot or dependence of the extracted weighted group velocity of polaritons on the actual spot size of the system. And you can see that in fact, even though we observe the change in threshold, so we observe approximately 2 times, not 2 times, 1.5 time change in threshold, which goes from 1.5 millijoules per centimeter squared up to 2.4 millijoules per centimeter square. We did not observe such dramatic, so I assume this, the word dramatic or strong, it is a very relative word. But we do not observe such big change in weighted group velocity. So it was less, so it was of 1.2 or 1.3, I think. And the weighted, I mean how it was calculated. So since we observe, since we see there is some distribution of polarity.
Sorry, just a second. Are you talking about figure 5.7?
Probably. Actually, I do not have the copy with me, but there are three or four insets in a row.
Because in that case, I also have a question on how you can work out the group velocity so noisy.
No, actually, it's not.
The dispersion is actually quite well fit with the parabola.
Of course, because this is how actually, yeah. Because above threshold, we can really easily. So let me, yeah, I've been given the copy of the thesis and I would like to comment on it. So because the figure, the panel B shows the fitted, so the central emission of the condensate that was obtained by fitting the cross sections along the energy axis or vertical axis. Yes, and that's why this curve looks really smooth, and it actually is the zoom out of the condensation region. But then also what we can extract from this fit is the intensity of polaritons, or like population of polaritons at each particular wave vector. But now when we calculate wave vector, it becomes a bit noisy, because in fact, in order to derive the group velocity, we need to use the derivative of the energy with respect to the k vector. And since there is small, of course, there are small peculiarities in the dispersion images you can see in panel A. So two adjacent points, two adjacent fitted points can slightly vary in values, and that's why it introduces a big noise into the group velocity. But still this figure allows, so the panel D and the panel C combined can allow us to calculate the weighted group velocity, because for each point on panel D, we know the weight, we know its weight from panel C. Now if we do the summation of all the points in panel D with the corresponding weights from panel C, normalized of course, we can find the weighted group velocity, and it has to come with error bars on the next figure 5.8. But to be honest, I actually for some reason I do not see those error bars on this figure, and yeah. But
right, and did you do the same for a bigger spot?
I did the same. You see, I did the same for 4 different spots. And actually, at the time of the analysis, this is so. At this point, I'm coming to the other comment that you made. Why the dispersions in panel C and F on the main slide of the presentation are not really symmetric? It is really the asymmetry in the experiment when it was measured. But for the analysis of group velocities, a very uniform and without any asymmetries dispersions were used. So from panel A, so they looked like from panel A, and for 4 different spot sizes. And you can see that the weighted group velocity actually rises. So it means that there is a spread. So polaritons tend to occupy states with higher and higher momentum once we reduce the size of the condensate. But the change of weighted group velocity is not really that pronounced as the change in the threshold values.
Exactly. So it's less than a factor of maybe 1.2.
Yeah,
right?
It's approximately the factor of 1.2. So it's I would say it's 25%. So yeah, 1.25. So we can see 5 and 4. Yeah. So it is increased.
And is this enough to justify the
but
change in
you
threshold?
can see these points are also stopped at 20 microns, but the threshold measurements were done even for smaller spots, so for 9 microns. So that's why I expect if we like extrapolate this fitted curve like black dashed line further, and
Okay, but you should
measure.
admit that if you look at the threshold from 20 to 40 is a straight line. You see that? I mean,
Yeah.
a part of your fitting, but it's a straight line.
It's
So all the physics is before 20 microns,
but
right?
all the physics is happens below, I would say 15 or 20 micrometers. That's where we observe the strongest.
And that's where you don't have the data for the group velocity.
And what?
And this is where you don't have the data for the group velocity.
And this is where, yes, unfortunately, I do not have the data for group velocity. But yeah, the assumption was that.
I mean, it's a good guess, but because I also don't know what else can we claim. Maybe some collective effect of all this filamentation, which is still some strange physics. So yes, I'm not criticizing, right? Your educated guess, but I think there is a bit of a lack of experimental evidence somehow.
Maybe yes, maybe there is some room for improvements, I would say, and for complementary measurements that would really, I would say, increase the quality and would prove some or gauge some hypothesis. Yes, but yeah. So, but this is what.
Sure, no problem. This is not a big problem. I mean, I like that actually there has been a strong effort to find a way, or at least find a reason on this. To give an explanation to this effect. Okay, let me go to another chapter. Like, this is some curiosity I have. You're not the only one to observe this blue shift. Could you go to the blue shift as a function of power that you have in, I don't know, the first experimental chapter? Maybe here it would be some.
You mean first experiment? Okay, so I'll go there. Oh, sorry, I went too far. So here, you mean?
Well, I don't see the blue shift, but.
So yeah, can you, Yelena, can you play please the video? So here,
Yes, here. Okay, so we know that there is a difference between inorganic and organic polaritons in terms of blue shift and what causes the blue shift, right? So I think there is much more an effect of quenching of the Rabi, right? So phase-space filling, I suppose. Yet for inorganic polaritons, the blue shift does not follow this strong non-linearity. So here it looks like the population of the polariton condensate is what is causing the blue shift. While we know that this is, well, for, I would say, both organic and inorganic, even considering the phase-space filling should be the reservoir that is actually giving this strong blue shift to all the polariton branch. So why do you think in this case, I mean, you are not the only one observing that, so I'm not saying that this is not a condensate. Why do you think this case is following the population of the condensate?
So
and not the pump power, let's say.
And the last part, what? Can you please repeat?
I mean, why do you think in this case it's following so much the condensate population rather than the reservoir, which is given just by the pump power?
So the question of the blue shift behavior and its origins were actually studied by my colleagues. So I have cited it here by Yegafarov, by Timur Yegafarov and the team. And there, so they outlined or came up with the explanation that there are two main contributions to the blue shift that happens in the inorganic systems. So first of all, first one, as you said, it is the quenching of the Rabi splitting, which happens due to the saturation of strongly coupled organic molecules to the cavity mode. Because inside the cavity we have the polymer. And this polymer encompasses molecules which are strongly coupled to the cavity and which are not strongly coupled to the cavity. And when we pump the system non-resonantly, we excite both of them. So upon increasing the pump power, we populate either the strongly coupled molecules and not strongly coupled, weakly coupled molecules, so we can put it this way. And the saturation of the strongly coupled molecules leads to the quenching of the Rabi splitting, yes. But the population of non-strongly coupled molecules, which amount is, I would say, dominates strongly coupled molecules, leads to the renormalization of the cavity resonance. And that can be seen from the reflectivity, not from the reflectivity, but it just renormalizes the cavity mode. And those two contributions are of equal effect. So they are they happen. So they contribute equally to the blue shift. And I think this actually answers the question, because so the blue shift itself was attributed to populating both type of molecules. That's why
okay, but this doesn't really answer too much, because the population of these molecules, so it's just excitons, right?
Yes.
Should be linear with the pump power.
But
where the pump power is going.
So
unless there is a sort of condensation even on the exciton.
Condensation even on the excitons.
No, I mean, I'm just saying that the exciton reservoir should be filled by the pump.
Yes, and at some point, at some pump power,
non-linear.
But at some power, it saturates, right? So we observe the non-linear. You mean why the blue shift? I see. Okay. Yes. If we increase the pump power linearly, then the blue shift should follow, as you say, the linear population of the excitons there, and not rather the non-linear population of polaritons. But this is okay. This is an interesting question, but okay.
Again, you're not the only
That's
one
what
that
we
is
actually, I
seeing
never
that.
questioned that part, because that's what we observe in all our experiments with any kind, with different kind of molecular or organic materials when we study it at room
I know
temperature.
that, sure.
Body pi, conjugated polymers, like G1 aggregates, they all exhibit this step-like behavior. But also what I also studied is I tried to do the dispersion images and the dependence on the pump power when I separate the TM TE splitting, because because the condensation in fact happens in those two modes, and it doesn't happen at the same time, as it turned out. That's why maybe if we cut out for example, one of the modes and just observe the other, we would obtain a smoother transition, because then it would be more linear. But it's this requires a bit more like investigation, because I cannot like really answer from what we have so far.
Okay, maybe another couple of questions. Look, you can go to the slide that also Simone was asking, and this was the time resolve dependence on the condensation. Yes. Okay. So here you are describing this. So you are excited with a let's say quasi-continuum laser compared to the cavity lifetime, right?
Right.
Because you use a 5.5 nanosecond pulse, while I suppose the lifetime of the cavity would be femtoseconds in this case, correct?
Correct.
Okay, but here you see a difference between the decay of the polariton population and the pump pulse.
Mm-hmm.
And you say that there is this, I don't know, it's one, I suppose is, I don't know, some nanosecond of decay time for the exciton, for the polariton. I don't remember what you said at this point.
Yeah, actually, the decay time. So the decay slope of polarity and photoluminescence in case of the below threshold, so the slope follows the slope of the pump pulse because it's linear.
Sure, but it must follow completely because it's much faster than the pump, right?
It's
faster
than the pump.
There is a strong delay in the relaxation process.
Yes.
But not the relaxation of the molecule, which is quite fast.
Mm-hmm.
So do you think this is due to a bottleneck effect?
Bottleneck effect.
I don't know, from exciton to polariton? Because otherwise I would expect
in this case,
time.
actually, the lifetime of polaritons, whether they are on the so they actually occupy the whole like polariton branch. The lifetime of polaritons is much shorter than the lifetime of the excitons in this case. That's why
sure it must be. It's like femtoseconds against
femtoseconds, yes, by several orders. In this case, 3 orders.
Exactly. So you are following what? Because of course, in this case, the reservoir is very long, right? But somehow it's filling the polariton state.
I'm sorry, I didn't get the last part.
Okay. So this is the lifetime of the exciton reservoir. You are saying,
yes.
Okay.
Mm-hmm.
Which lasts longer than the pulse somehow.
No. So there is a small delay between the photoluminescence intensity and between the pump pulse. So if we assume that, and actually below threshold, the photoluminescence is linear with respect to the pump. That's why when the pump, when we decrease the pump, there is still some residual photoluminescence that can be attributed to the previous moments of time, and this delay along the vertical horizontal line is exactly is exactly the lifetime of the excitons, because as I told previously, if we excite the system with a delta function, we would obtain so the decay, the photoluminescence decay, which is which full width at half maximum is around 500 picoseconds. And this full width at half maximum sort of must coincide with this time lag between the pump pulse and the photoluminescence pulse.
Okay.
Did I answer your question?
Sure, you did. Okay, fine. Okay, now I'm curious on this new structure that you have proposed, which is a hybrid
It was,
metal.
I want to make a point that it was not proposed by me, but by our colleagues in University of Sheffield.
No, sure. Okay. I don't know, but you studied it, right?
Of course, yes.
So somehow the aim of this new structure is to have let's say better to some extent a better cavity than the usual DBR, right?
No, the aim was to actually simplify the fabrication process, but to keep the parameters of those cavities on the level compared to the conventional DBR structures, not to exceed those the quality of the, but just to demonstrate.
You actually start, at least in the thesis, describing this in a very positive way, which I do agree could be it could be actually like you say. Let me see the page of this. Okay, maybe the best is figure. Maybe you have there in the presentation. It's figure 4. I don't remember. It could be 18. Here. Okay, perfect. Okay, so to some extent, you have a Q which is increasing even beyond the DBR structure. So this is quite interesting, right? And at the same time, you mention also in the thesis that the penetration length is actually reduced due to the very small pairs that you put before the mirror. So the penetration length of the optical mode in the cavity is much shorter.
For the higher number of DBR pairs on top of the substrate, Argentum substrate, yes.
No, well, I'm just talking about the bottom DBR or the bottom structure, hybrid structure. Okay, so on the one hand, you say that this is the case, and also in the new version of the thesis, you show a model in which this penetration gets, let's say, larger and larger, increasing the number of pairs.
No, actually, the penetration becomes smaller and smaller once we increase the. No, it becomes. Sorry.
I think it becomes larger because you've got more pairs, right? So.
Yes, it becomes
the
larger.
cavity is always in the same place. However, here it seems that if you look at the panel B, it seems that the H1, of course, has got a very, very small penetration, let's say 16 nanometers. But then I go immediately to 120. And also in terms of line width, so the Q factor, which depends on the line width though, seems to increase very much, increasing the pairs and even going beyond exactly the DBR structure. However, the line width already at H3, it gets kind of smaller, but then it stops. It doesn't increase more. So why there is this discrepancy compared also to the theoretical structure, let's say?
So actually, the quality factor of the system is the function of different parameters, and if the line width so the quality factor of the cavity just defines how good the resonance is, and but the line width, okay, this is interesting. Yes.
Also, you have a Rabi splitting, which is supposed to be bigger because the mode has got a smaller volume. And this is actually, even experimentally, is following this behavior. So it seems that the penetration length is actually not increasing so much at the beginning because the Rabi is still high. So you see this red line?
So red line, yes.
So given that this is experimental, I believe it somehow. So to some extent, you see this reduction of the volume, which is good because you've got a higher Rabi splitting, for instance. But why you don't see this in the penetration length? I don't know how you got this penetration. Is this theoretical?
This is the theoretical yes curve, and yeah, we didn't actually conduct it.
It seems strange, right? Because it should be much higher than that.
Yeah, increasing the penetration,
much smoother, let's say.
Length into the bottom mirror should, I would say, increase the mode volume. Yes, increase the mode volume of the cavity, and it should result in the reduction. And it should result in the reduction of Rabi splitting. That's what we observe. But why the quality factor still increases? But the quality factor, so the sharpness of the resonance that we have in our system highly depends on the reflectivity properties, and this is just the So yeah, I'm actually struggling
Anton,
to answer
I
it.
guess, can I just enter the discussion? I guess that penetration depth is a matter of definition. For instance, imagine you have exponential decay of your field. Yeah, then penetration depth will be constant, but of course the quality factor will be controlled by the exponential tail. Yeah, in this sense, you will have the same penetration depth formally, but exponential decaying contribution to the quality factor. I guess it is more or less what you see here. The fact that you do not change the penetration depth, but by adding additional mirrors, I think it doesn't mean that you do not change the quality factor, because anyway, by removing your metallic absorbing layer far and far, you increase the quality factor. I think it is what you see. Dear colleagues, I just want to say that already 1 hour putting the question.
Okay, sure.
Sit in the way we will overlap the next defense.
But the
discussion is interesting and constructive, but we should look in future. And as a chairman, I just want to politely ask you to somehow follow the timetable. Okay.
But thank you, Daniele, for your question, because it's really, so I will revise this.
Thank you.
Yeah, thank you.
Great. Daniel, do you want to ask some more questions? Or if there are urgent one?
This is fine. This is fine for me. Thank you.
Great. Thank you very much indeed. Dear colleagues, then let's go on. Who from Skoltech members would like to be the next question person?
Yeah, Nikolai, let's start, yeah?
Yes, certainly.
Okay, thank you so much. First of all, I would like to say...
to restrict the time of questions.
Yes, of course. Don't worry, Nikolai. Yeah, okay. First of all, I would like to say that I really like this work. And Anton answered all of my questions, and he has done corrections, agree with my suggestions. And thank you so much for this, and I'm really satisfied. And but I have some technical questions. The first one related to the slide number 10. And we see there are many of organic molecules on these slides, and Anton chose 2 of them, yeah? And I would like to ask to formulate maybe some requirements to the organic molecules that after that we can use these molecules as optimal one for such type of experiments. Is it possible or not to formulate this list of requirements to organic molecules?
Okay, thank you. Sorry. Thank you for your question. So, in fact, okay. The question of which organic molecules and which, for example, which fluorescent molecules are suitable for the polarity and in particular applications is a very
correct.
I'd say difficult question, because a lot of groups has done a lot of work and have been working on it for many years, for exceeding like 5 years to find the appropriate and photostable material that could really be coupled to the cavity mode, and that could sustain and could avoid the photodegradation that is really a challenge when we excited with the high fluencies. And so far, different fluorescent molecular dyes have been studied before me, and even by my colleagues, for example, Timur and Denis Sannikov, so in particular the G aggregates. But each time when you use or choose another molecule to test it, it is really an unexplored, I would say, an uncharted territory, because you really need to go with the first run. So you put it there and you see whether it gives any promising results or not. But so far, I do not have like that vast experience across other different molecules from the experimental point of view, because I work predominantly with this brominated fluorescent molecular dyes and conjugated polymers. So this is I think I will not be able to formulate the list that you. So, but the main criteria for the dyes is that they possess fluorescence, so they can shine. But so for the next steps, it's really, it's really. I didn't do the fabrication myself, so thank you.
Okay, what did you do for prevent of photodegradation in your experiment?
So usually during the fabrication stage, in order to prevent the photodegradation, there is an additional slab which people place in between the active medium and the surface of the bottom of the DBR layers, which is silicium O2. It prevents the contact. So this lab actually is the attempt to prevent this photodegradation that usually happens when the medium is excited with the optical excitation. And in our experiments, we try to reduce. First of all, we try to operate in the single shot regime, and do not exceed the critical pump fluencies from where the polymer is damaged completely, so it can no longer it is damaged, so it is burned out. So from practical point of view, you cannot really do much or a lot once you have the cavity. You can really just find the appropriate parameter ranges where you can safely operate without any effects of photo degradation. And mostly it is the length of the pulses, peak energy of the pulses, and the overall fluence that you that you send to the sample, and the repetition rate. And that's it.
Okay, thank you so much. And the last question related to the slide 17. Yeah. Okay, the idea is, did you try to evaluate, or do you know the concentration of your dye in active layer?
So actually, it was not. Yes, and I think it should be written somewhere in the thesis.
Yeah, but the question is, did you optimize this one, or your colleagues who prepare the sample, or what do you think? Why I have asked this one? Because fluorescence properties depends really on the concentration of this dye. For example, we can obtain self-quenching concentration and so on.
Yes, and actually you're very right. I had different test samples with different concentrations of this molecular dye in the polymer, and so from my head, I cannot really recall what was particular concentration for the successful cavities, but the concentration cannot be really high, because that's where I didn't observe any lasing at all or any condensation. And yes, there is for sure the critical concentration and the optimal concentration of the molecules, and it depends. And it really depends on the particular dye. Yes, you put or you insert in the active in between the DBRs. And for this, it was around. If I'm not mistaken, it did not exceed 10%.
Mm-hmm.
Yeah, that's it. But I can recall later. Mm-hmm.
Okay, thank you. I finished, Nikolai.
Thank you very much, Dmitry.
Thank you.
Skelaris, it's your turn now.
Anton, thank you for the very nice presentation. I have a couple of questions. The first question is, in the whole of your work, you've been using a pulsed pump, right? What would happen if you use a CW pump? Will you be able to observe lasing? And if not, why?
I believe so. To be honest, I never tried. All the material that I worked with under CW excitation, but I believe that the threshold for the CW operation, for the CW laser, in order to absorb the condensation, would be much higher, because the average energy, or the peak, the average energy in the pulse is lower. That's why you need to pump your system higher. But due to this fact, you will not, the damage threshold of the organic material inside you will reach it sooner than you will reach the threshold for the condensation. That's why we use the pulsed excitation, because the peak intensity is much higher, and you can really instantly create the population, of excitons, which will sooner collapse to the polarity and condensation. I think.
So the damage is a thermal damage then?
Thermal damage, yes.
Okay. Now, as an expert in this ambient polaritonics, right? So
Right.
basically, you're trying to transfer all this. No, you are the expert, not me.
Okay. Understood.
So you're trying to transfer all this know-how from the inorganic cryogenic temperature regime to the room temperature regime, right?
Right.
So what, in your opinion, is the future for ambient polaritonics? Is it organics is the future, or some other inorganic materials that would operate?
So I believe actually that the time for, the time has come for organic polaritonics, and in particular, there are perovskite structures that exhibit a really good, so aggregate the best properties of organic materials, which, for example, their ability to operate at room temperature, and also the structure of the perovskites is of the same level as the inorganic counterparts. That's why I believe that the fact that you don't need the cryogenic setups in order to observe all this peculiar interaction between the condensates that was observed and that has recently been that has been studied a lot in organics fields. It could be so. It is potentially, and actually, it is now being transferring to the organic materials. And yes, I think the future, if we are considering just the future of polaritonics, I think the future lies somewhere in between, where we can integrate some advantages of organic materials, because it cannot completely replace the inorganic part, with the inorganic ones. So, and some intermixing between them, yes, would really make some, I don't know, but it's really difficult because usually we operate at different wavelengths, so there must be found a way of bridging the organic and inorganic one. But now the organic polaritonics with perovskites is coming to the level of inorganics. That's why we can really, with the second one, contribute to the inorganics, and hopefully vice versa. But yeah, these are mainly like fabricational challenges, and because yes, all the apparatus for the experiments has been already developed, and but it requires some modifications here. So this is really the fabricational challenges, and there are a lot of them.
Thank you.
But I believe we will overcome them.
That's me, yeah.
Thank you very much, Sigilaris. Okay, Anton, I have just two short questions. First one is continuation of the question of Sigilaris. He asked you about CW direction, and I would ask you in the opposite direction. So why you are using nanosecond pumping, not, for instance, picosecond?
No, actually, usually we use femtoseconds and picoseconds, but nobody has seen the condensation under the nanosecond excitation due to just these particular reasons. And that's why for this first experiments, or the first chapter of my thesis, I used nanoseconds just to test how far we can go and how long and what, so how long we can, so what condensates and of what maximum lifetime we can obtain, and find the conditions that we can maintain in order to operate under like CW excitation. Of course, quasi CW. Of course, nanoseconds is not CW, and it is far from the ideal. Yes, but still, it is a good. So I would say a big leap forward towards this direction. So even though it is just nanoseconds, but usually, yes, usually we work with femtoseconds and picoseconds, and like with this pulse duration, samples can really sustain thousands and millions of pulses, especially the conjugated polymer. So that's why it's really suitable for studying some statistical properties and other physics that require it to be prone to photodegradation. Yeah.
Thank you very much. And one more question about, you mentioned disorder in this structure of your condensate. So if I understood correctly, the disorder itself, it is the same from pulse to pulse. Yeah, I mean the landscape disorder.
So the potential landscape is is
I
the
just
same.
haven't finished. So my question is: these filaments of the structure of your condensate, how random is it? Is it reproducible from pulse to pulse?
It's different from pulse to pulse. As if you just compare one realization to the next realization. But if you average over many realizations, you can see that there is. So you can picture it this way. So there is some static disorder, some static pattern that is different from position to position. But on top of this static pattern, you can still have a stochastic disorder, which is on top of this static picture. And that's what we can observe from single shot measurements from pulse to pulse. But if we average over many realizations, we sort of we draw the static pattern that is also present there, and the different reasons for the static pattern and for the stochastic one. So,
and this stochastic pattern, it comes from what? Excitation stochastics or some condensation stochastics?
So I would say it doesn't come from the pump profile, because the pump profile is really smooth and homogeneous from pulse to pulse. It's really consistent in time. So I would say it comes like there are different initial conditions in terms of the noise in the system. And that's why from pulse to pulse, you can really create sort of like different systems where like different instabilities, they also come into play, and it modifies the potential like stochastic landscape. But so this is a bit speculative answer, because it needs to be really studied a bit more in detail. So
what you answered clearly, that it is not from the excitation. The excitation,
it is not from the excitation.
Perfectly.
Yes,
it
is.
Is random,
yes,
okay.
Because we confirmed it at the very first stage of the of the experiment.
Thank you very much for a clear answer. I also satisfied with all your modifications. And now let me ask the audience if you have whatever questions you want to raise now, discuss now. I don't know. Yes, please, Sergey.
Can you go to slide number 9? The question is about line widths. If you play the video, at 2 times P threshold, you have practically the same line widths as before threshold. Can you comment on this? And does it mean that the system loses coherence properties?
It actually, so we usually attribute it, no, it doesn't lose the coherence, but it is due to the dynamical blue shift that we can observe actually on the top left panel. Just because the condensate undergoes the blue shift, and which is keep on increasing upon increasing the pump power, we observe this sort of smearing of the line widths. But no, but since I told you previously that I did not study in particular the first order coherence of the system with respect to the other parameters of the system, I cannot tell you how much it loses in the degree of the coherence. But it is still the condensation, and it still has the temporal coherence, and so it doesn't.
Thank you. And one more question. At slide 24, you have measured statistics over 300 shots. The question, is it a good number? So definitely it is enough, but the question, isn't it too much to already create some deterioration in your sample? Did you check how many pulses you can send to?
Yes, thank you. I understand your question. This number of pulses is not enough to like induce any kind of deterioration on the sample, because these are conjugated polymers, and we have shown that they can sustain millions of pulses without any without altering the emission properties. And this is why with this sort of sets of measurements, we can really obtain a lot of data sets without changing the statistical properties. And that's what was shown, yeah. But was not actually included in this work, but was mentioned in previous work on the same material, yeah. Thank you.
Yes, please.
Yeah, Anton, thank you for your talk. So I have a question regarding the G2 measurements. So the most common way to measure G2 is HBT setup with TCSPC and APDs. So I wonder if you tried it for your system, and do you think it is applicable method for your?
So the answer to the first part is not. I have not tried the HBT experiment in the organics, and I think it is applicable, and it actually would be a really good experiment to check whether this method, so in order to compare those two methods, because we know that for the HBT measurements has a finite like time resolution, which is of the order of 40 or 30 picoseconds. And here in our case, we have a much better resolution, which is defined by the lifetime of our polariton condensate. But this is a very interesting, and yes, this experiment would clearly definitely clarify the picture and would provide very insightful information, to compare it with, because until now only HBT experiments have been used to measure, but at least a 0 time delay. Yeah.
Okay, I see. Yeah. Thank you.
Okay.
Thank you very much.
Thank you very much, dear colleagues. Very interesting discussions. If there are no more questions, then
We actually do have one more question online.
Online, perfectly.
Yes.
Please.
So, Anton Zasidatelyev, please, could you unmute?
Can you hear me?
Yes,
Yes,
Anton, thank you.
Okay, I'm not sure if you can see me, but I hope it is. You can hear me clearly. First of all, thanks, Anton, for a nice talk. And it's really very, very cool to see you achieving this milestone after many years working together. It's great. Of course, I'm familiar with most of the stuff, and just maybe one part is not very, which I wasn't really participating. Is about DBR, metal DBR mirrors, and I think it's slide number 18 is where I have a question. If you can move there.
Can you please bring back the presentation? Slide number 17.
18. 18. Sorry.
18. Yes, this one. Okay.
No, I can't see slides.
No, just put on the presentation without this full screen mode. Yeah. Okay. Yeah, here.
Okay, great. I was wondering why at the metal DBR structure you have way broader K distribution with respect to DBR. Could you comment on this, please?
So that was an experimental like evidence, and actually, so the conditions of the measurements were the same. So the spot size was the same, and but that's what we observed, and that's what actually I don't think we have a correct or exact or answer that we really agree upon altogether. But I don't know. This is what we observed. We observed that in K space we also have a broader distribution in K vectors than.
So is it actually the same power for the?
It is it should be the same power for panels B and B with respect to the threshold for two different cases. But yes, that's what So with conventional DBR cavities, and so in this experiment, I mostly observed the collapse to the ground state in DBR cavities. But in most cases in metal DBRs, I observed that it has it has a broader distribution in K-space. Yes, that's.
I was just wondering, could it be attributed actually to the twice larger the blue shift that you have there, or could it be the effect of kind of nonlinear repulsion or some effective?
It could be actually, yes, because what we observed previously, yes, that so in the consequent work, but it is construction that so the blue shift goes side by side with the increasing or dynamical blue shift actually also causes the spread in K-space and group velocity. So here, if the blue shift in the metal DBR cavities is more pronounced, then then it might be that this is the main cause why we observe the more spread in. But also what we also see upon on top of the wider K-space distribution is a small, like I would say, fringes on top of the on top of the emission in K-space. So, but I think this is a different story. This is just this might come from some sort of the interference between the emission. But yes, it might be due to the fact that we observe a strong blue shift there, so it's almost 2 times stronger.
Thank you. And can you jump, please, to the slide Daniela has had a question about the group velocity? I just have a quick comment there.
Yeah.
Yeah, here, yes. I actually believe that the main strong dependence on the threshold comes from 1/r contribution here, but not with the group velocity contribution. Even if you put the constant group velocity, the strong dependence on the threshold is just because 1/r dependence. So basically, reducing the size, you increase the outflow anyways. So this is the biggest effect, I guess. Just a comment. Yeah. And just another comment. I think that actually the jump in the step-like behavior in the blue shift comes from the intermolecular interaction that we showed previously. This is just by the fact that the excitons are getting redistributed below threshold among all possible projections of the dipole moments. But above the threshold, it is not the case because above threshold, you have stimulated condensation, which is very faster than the intermolecular interaction. It's another comment. Thank you. That's it.
Good.
Thank you.
Very interesting comments. And probably we should stop discussion at this point. Do we have some other comments from the audience? No?
No, I can't see any
Dear
other
colleagues,
comments.
then I should announce that we are coming to the most beautiful stage where it will be not the questions, but I hope statements of the supervisors. And I propose to start with Pavlos and proceed with Denis. Okay? Pavlos, can you say some words now?
Thank you, Nikolai. I hope you can all hear me. So it is really with great pleasure that, Anton has come. I've seen Anton to come to this stage, really starting from a completely different background and making a strong contribution to the field of polaritonics and becoming what I would say an expert, as you could judge from his, both from the quality of the presentation and the results, but also from the scientific debate that he can hold on the results that he has obtained. So it was really a wonderful experience working together with Anton. He has his own point of view that he can defend, and in many cases it happened that he was right. He could convince me otherwise to my original intuition on the experiments that he was doing. And I hope that actually in the time to come, we can continue to work together and bring to completion the several papers that he has submitted or about to be submitted. So Anton, thank you very much for all the effort that you have put in the group, which really shows in these wonderful results that you bring here today.
Okay, thank you, Pavlos.
Thank you very much, Pavlos. Denis, it's your time now.
Yeah, we work with Anton for a couple of years, and I'm proud to work with such a good PhD student, which I believe become a PhD. And I want to say that Anton is a really self-sufficient scientist, very robust, and he work alone. He can make good progress in the work he did, and he do not require any help. And yeah, that's all. I can make a statement that he is a very good scientist in polaritonics. Yeah, thank you.
Okay, dear Pavlos, dear Denis, thank you very much for your great contribution once again. And now let's follow instructions from Yelena, because we should proceed with deliberation.
Yes, thank you, Professor Gippius. So we are moving to the close part of the defense, which is deliberation and voting only by the jury members. So we kindly ask Anton and all other audience members to leave the room. For those who attend online, you are most welcome to stay online. We will open a separate breakout room only for the jury members. So Professor Deliberata and Professor Sanvito, please join it when you receive the invitation. And all the rest of the audience are most welcome to stay online and just wait for the announcement of the result.
Great. So, dear colleagues, after long and heavy discussion, we have to come to the decision that thesis is accepted as is. And I want to congratulate Anton and his supervisors.
Okay, thank you very much. So thank you everybody who was able to make it today to this very important date for me and for, yes, I've been, it was a long journey, actually 4 years of a really hard and I would say exhausting sometimes work with a lot of struggles and obstacles that I did not, so I overcame not alone, but thankfully to the team that I worked with in side by side, and especially to Denis Sannikov, to Anton Zasidatelyev, Pavlos Logudakis, and also the rest of the group, so Sergey Alyatkin, Ivan Gnusov, Stepan Barashov, Kirill Sitnik, and all the rest of the group. So this was, I would not be able to make it without long discussions on the physics themes and questions, and not only on those, with all the jokes and all the late time conversations and coffee breaks. Yes, so thank you very much, jury members, for really providing me a very constructive and like straightforward feedback, which really improved the quality of this work. And yeah, this is always this sort of feedback always puts you in the place and makes you to really digest and to improve whatever you do to further. And it is always yeah, you can always wish higher standards. And yeah, and thank you very much. So that's it, I think, that I wanted to say.
Okay, Anton, thank you very much once again. It's a great day for you. Thank you very much.